Okay, we're going to be talking about T cell development. And remember, T cells are born in the bone marrow, that's where they're created, and then they move out of the bone marrow and they move to an area through the blood and they move into the area of the thymic cortex. Thymic cortex in the outer region of the thymus gland. And this is where they're going to get educated. So let's just start with the hematopoietic stem cells in the bone marrow can become any kind of blood. They can become white blood cells or red blood cells. Well, these become precursor cells that are pre T cells uh, or pre thymocytes, and they migrate using specific receptor markers on them to know when they get to the right place. Because remember, they don't have brains or eyes or anything. So it's when those chemical signals build up and they put the right kind of surface markers on there, like c-KIT, for example. Then when they get there, they stick in certain areas. It's just where they're supposed to migrate to. So now these T cell precursor cells at this point are called double negatives because they do not express either the T cell for the CD4s or for the CD8s. So at this point, they're going to go through a lot of uh, TCR, T cell receptor, beta locus rearrangement and proliferation. So they can be multiplying it throughout these steps right here. So that means that once you get one that has rearranged successfully the TCR, you can get multiple copies of it with slight variations, and then they can all move into the double negative phase, which is where they have now committed to be a T cell. They don't really start expressing or proliferating a lot until they get into the double negative 3 stage, and that's where beta selectin is able to act upon those cells. Then you have double negative 4. This is right before they become positives. It's TCR alpha locus. They're going to begin producing the T cell receptors, both for CD4 and CD8s. And at that point, they become double positives. So they haven't decided what they want to do yet. This early development is going to be occurring within that thymic cortex. And then the positive and negative selection is only going to be occurring after you reach the double positive stage. And what do they mean by positive and negative selection? Well, positives, remember when T cells and B cells and all come together, they have to answer two questions. Are you self? If the answer is correct, then you get to live. Are you carrying self antigen? If the question is correct, then you get to live. So, what happens here is that you have these double positives that haven't decided what they want to do yet, but they can go through differentiation here. So if neither of the CD8s or CD4s are able to interact with M8Cs that are found on the surface of epithelial tissue lining this thymic cortex and thymic medulla, then if neither one of them are able to react with those M8C markers, then they will go through automatic apoptosis or death by neglect because they were unable to recognize self MHC. So you want to recognize self MHC. But there's also an issue of overreacting to MHC. If you overreact in the thymus gland to MHC, that means you're going to attack the cell containing that. And those cells will be initiated into death because of their overreaction. Most do not. And a lot of this is somewhat random. I mean, there's evidence they were able to use a cell sorter to filter out the CD8 cells from a guinea pig. I think it was a guinea pig, but from some animal, and and put the CD4 cells back in. So the animal had the normal number of CD4s, but they had lower numbers of CD8s, and the thymus started spitting out higher numbers of the CD8s over the CD4s because that's what was needed in the animal at the time. They really don't understand the concept of how that works yet. Um, well, I don't understand it, and I've not read any papers that suggest they do. Um, so it seems that the body has the ability to select the ratio of CD4s and CD8s that are being produced. Um, so what happens is after they interact with an MHC, whether it's an MHC class 1 or MHC class 2, will determine which type of T cell they're going to become. So now they're going to be called single positive cells. Um, and at this point, they're also going to be tested to make sure they're right in this point in here. They're going to be tested to make sure they're not overreacting to self antigen. That's something that we'll talk about in a different slide, so I won't go into too much detail on it. But if they're overreacting to self-antigen, then that would cause autoimmune diseases. At this point, you have naive T cells, naive CD8s and naive CD4s, and they can either be activated within the thymus gland or they can be secreted. They go through the thymic medulla, enter the bloodstream, and migrate out. Usually, the naive T cells will move to thymic tissue. So they go through the afferent blood vessels into the lymph nodes. They hang out there for a while and they'll be activated if they come into contact with self-MHC carrying the antigen that will stimulate them to go into clonal expansion. That's in a quick nutshell how that works. There's some other lineages like T regulatory cells, which are upregulated in the case of certain types of cancer and certain types of virology. Natural killer cells also can go through thymic selection. And this is, although there's been quite a few papers on it, it's something that we're not going to talk about in this class too much. There's a huge amount of research if you're interested in cancer research. T regulatory cells, being able to control those cells could actually be a way of treating a lot of different types of cancers. Because if those cells are upregulated by a virus or by cancer, what they wind up doing is downregulating other T cells and preventing them from having activity. So if you've got cancer and you could go in and you could neutralize the T regulatory cells for your cancer, then it will increase other cytotoxic T cells specifically, increase their activity against that type of cancer. It's a fascinating area of study that's going on right now. But again, all of these can migrate out into peripheral tissues. Remember, this is a very quick overview. You need to read the chapter. I really do not have time to sit here and read every single sentence out of the book to you. So I'm just giving you quick overviews in some of these videos. They've worked with how bone marrow stem cells become certain types of cells. And they do that because of interactions with stromal tissues or stromal cells, the yellow fat that's found inside the bone marrow, and along with other growth factors. So interleukins and specific receptors that are found on the surface of the stromal cells, plus actually just kind of squeezing them through those, will determine whether or not they become Notch positive exposure will cause them to become T cells. If they're exposed to Notch negative cells, stromal cells, then they'll become B cell lineages. And that can vary depending on the signal and types of interleukins that are there, but this is a basic way that they determined how that works. So they're doing a lot of work with stem cells now, and they're better able to take a pluripotent stem cell, duplicate it, and then cause it to differentiate into certain types of cells. So that may become a route of treatment for certain types of cancers in the future. If you could find a pre-B cell or even a naive pre-T cell or naive T cell that is specific to a certain type of cancer, and you could cause it to go through clonal expansion or increase the numbers of them and harvest those cells and put them back into the patient, then you've just declared war by sending a huge army to overwhelm the cancer cells. And last but not least, I'm not going to talk a lot about this, but the way the double negative thymocytes develop within the thymus gland and within the body has to do with whether or not they're producing certain receptors. CD117 is also known as C-Kit. It's one of my pet ones that I like to talk about. And it's remember the way the signal transduction works. It's the fact that you have the C-Kit produced means you are getting a signal. If you have it produced in high concentrations, you're getting a strong signal. If you have it produced in low concentrations, you're getting low or no signal. So that's how a lot of these developmental processes work. Take the double negative ones. They have no T cell receptors on the surface. They're found in the bone marrow and they will migrate to the thymus. The C-Kit, the CD117, is in high numbers on the surface of the cells, as is CD44, but they do not have the CD25. So they migrate through the bloodstream, and these receptors on the surface allow them to adhere to thymic tissue in the medulla cortex. Then you have the double negative cells, which have both the C-Kits, CD44s, and they've brought in a new player here at CD25. So they move into that subcapsular cortex, kind of underneath the very beginning of it. So then they are starting some of their gene rearrangements because the CD25 will interact with whatever triggers will bind to that. At this point, you start seeing a reduction in the C-Kit, and the CD44 is non-existent. So they moved into the subcapsular cortex, and you start getting pre-T cell receptors and B selectins on the surface. These are pre-T cell receptors. They are not T cell receptors yet. C-Kit, very low concentration. CD44 is non-existent, and CD25 is non-existent. So now you can kind of think of these as being able to go in and you hang on at the top of the ladder because you have these two. And then you hang on for quite a bit of time in this subcapsular cortex because you've got all these little hangers on there. And then you start losing those and you start falling down through the netting, through the ladder, through the scaffolding, however you want to imagine it, until you get down into this subcapsular cortex, to the cortex region. And that's where you're going to get proliferation of and allelic exclusion of B chains, beta chains, and alpha chain locus rearrangements. So you can become double positive thymocytes at this point.